All right, let's dive right in. Today, we're tackling a really fundamental question. Where does cancer actually come from? And the answer might surprise you. See, it's not some kind of foreign invader. The real story of cancer starts deep, deep inside our own cells, with the very genes that are designed to help us live.
So, to get our heads around this, let's start with a simple idea. Think about it like this. Every single one of your cells has a kind of accelerator pedal, a growth switch that tells it when to divide. It's totally essential for, being alive. But what if that pedal got jammed down? What if that switch got stuck in the on position? That's really the heart of what we're talking about today.
And to understand how that switch gets stuck, we have to go right back to the beginning, to the genetic origins of cancer. This isn't a story about some outside force. It's about a breakdown in our body's own programming. When you strip away all the complexity, the definition of cancer is really about two words: uncontrolled and abnormal. Your body has these amazing, incredibly precise systems for controlling cell growth, for telling cells when to stop, and even when it's time to, die. Cancer is just what happens when those built-in safety features fail. So, what causes that failure? 9 times out of 10, it all boils down to damage. Specifically, damage to our DNA, our genetic blueprint, that for some reason just doesn't get fixed. That's the moment the mistake, the mutation, gets locked in. Okay, so before we dig into how the system breaks down, we really have to appreciate how it's supposed to work. It's actually a beautiful system. Our cells have this whole team of genetic guardians that are constantly working to keep everything in order.
Think of it like a 4-part security system. You've got the accelerators, right? The genes that say go. Then you've got the brakes, the genes that say stop. You've also got a self-destruct mechanism for cells that are too damaged to fix. And finally, you have the DNA repair crew itself. For everything to work, all 4 have to be in sync. Today, our story starts with that first one, the accelerator. These accelerator genes have a name, Proto-Oncogenes. And listen, this is so important to understand. These are not bad genes, not at all. They're actually the heroes of the story. They are the essential go signals for growth, and they're crucial for everything from how we develop as an embryo to how we heal a paper cut.
Exactly. I mean, this quote really says it all. These are good and beneficial genes. They literally build us. But here's the catch, and it's a big one. Once their job is done, they're supposed to get quiet. The problem starts when they just don't.
And that's where the transformation happens. How does a gene that is so helpful, so necessary, become the driving force behind cancer? This is the exact moment our hero gene starts to go rogue. So this whole transformation gets kicked off by damage, usually from things we call carcinogens. And what's really fascinating and kind of scary is how specific this damage can be. We're not talking about just random chaos. The chemicals in cigarette smoke, for instance, are known to cause a very specific typo in our DNA, swapping one chemical letter, a G, for a T. Sunlight does something similar. These tiny little mistakes can have huge consequences.
And that typo is what turns a proto-oncogene into an oncogene. The normal, helpful gene becomes a mutated, harmful one. The controlled, finely tuned accelerator becomes an accelerator that's just stuck to the floor, screaming, "Go, go, go," to the cell, leading to that out-of-control growth. Okay, so we know a proto-oncogene can be damaged and turned into an oncogene. But how? How exactly does that damage actually activate it? What's happening on a molecular level to flip that switch? Well, it turns out there are 3 main ways this corruption can happen. The first is called translocation. The gene literally gets snipped out of its normal spot and pasted into a new one, a hot spot on the chromosome that's basically always turned on. The second is gene amplification. This is when the cell's copy machine just goes haywire and makes hundreds of extra copies of the gene. And the third is that point mutation we talked about, that single-letter typo that creates a supercharged, hyperactive version of the growth protein. But here's the thing. It doesn't matter which of those 3 things happens, whether the gene is moved, copied, or mutated. The end result is exactly the same. The cell gets flooded with way too much of that growth signal. The balance is completely destroyed.
So let's make this real.
Because these aren't just abstract ideas in a textbook. These exact genetic screw-ups are directly linked to some of the most common cancers we know. It's time to meet the rogues gallery.
Just look at this table. This shows you the straight line from the mechanism to the disease. Take the MYC oncogene. When it gets moved, that's translocation, it's tied to Burkitt's lymphoma. When the ERB2 gene gets copied over and over, that's amplification, it's a major driver in many breast cancers. And when RAS gene gets that one little point mutation, it's famously connected to pancreatic cancer. It's a direct link.
That table shows the variety, right? But the story gets even more focused. In fact, the impact of just 2 of these rogue genes across all human cancers is, well, it's absolutely mind-blowing.
Let's just take the RAS gene family. That one specific error, a single-letter typo, a point mutation, is found in an estimated 30% of all human tumors. Just let that sink in for a second. Nearly a third of all cancers can be traced back, in part, to this one specific stuck accelerator problem.
And if you think that's a big number, then there's MYC. Its fingerprints are everywhere. In some way, shape, or form, the MYC oncogene is involved in an estimated 40% of all human tumors. Between just those 2 genes, you're looking at a huge slice of the entire cancer landscape.
Now, it's really important to remember this. Turning on an oncogene is a huge step. It's often the first big step, but it's almost never the only step. As all the research shows, developing cancer is a journey. This stuck accelerator is often just the very first domino to fall in a much longer, more complicated chain of events. Which leaves us with a really critical question, doesn't it? If getting the accelerator stuck is enough to kickstart all this chaos, what about the brakes? What happens when the other genetic guardians, the ones whose whole job is to stop a runaway car, also fail? Well, that's where the story gets even more interesting. Thanks for joining us.